Good day, everyone, and welcome. This is Ray Ford with FDA Center for Drug Evaluation and Research Small Business and Industry Assistance Program, also known as CDER SBIA. Thank you for joining us today for our presentation on FDA Study Data Technical Conformance Guide. The webinar will be posted in its entirety, both video and audio, on our website within 5 days, and that website is fda.gov/CDER SBIA webinars. Before we begin, I'd like to cover some details. Slides for today's presentation are available from the right-hand side of your screen where the orange button says Download Presentation. This activity has been pre-approved by reps, is eligible for up to 1 credit towards a participant's RAC recertification, and SQA approved for 0.25 non-GCP or non-GLP units per 1 hour towards RQAP re-registration. You'll be able to obtain the attendance certificate only upon completion of the survey, which will remain open for 2 weeks. That means it will close on December the 11th. Even if you don't need the certificate, we'd appreciate it if you let us know your comments and feedbacks via the survey. To outline today's event, the webinar in its entirety will run approximately 1 hour. Our presentation will be followed by a Q&A session. We'll now introduce our speakers. First, we have Helena Sviglin, MPH, from the Center for Drug Evaluation and Research, and Elaine Thompson, PhD, from the Center for Biologics Evaluation and Research. Joining us on our panel, we also have Ron Fitzmartin, PhD, MBA, from CBER, Virginia Hussong, also from CBER, and OBI Director Ethan Chen from CDER. Welcome, Elaine.
Thank you. Hi, everyone. This is Helena. I want to thank you all for being with us here today for this seminar where we will go over pertinent updates in the FDA Study Data Technical Conformance Guide version 4.2 that we released in October 2018. The topics we'll cover in this webinar will go over some of the new content for version 4.2, including all the appendices, the new parameter codes. We'll go over the new TAUGs, the Therapeutic Area User Guides, otherwise known as TAUGs, protocol deviations, and then we'll turn to Elaine for some updates to our SEND content. So as you guys know, as you may know, we've been publishing a technical conformance guide starting in 2014. We publish one every March and every October, or we intend to. We've never missed a deadline, and we will occasionally put out a special edition if the content is pertinent enough. So this is our October 2018. Just past the title pages in the document, you'll see a revision history for each version that we've put out. Here is the one for October, and the areas highlighted in gray are the topics that we'll cover in today's webinar. So for the new content, we'll start with the appendices. You guys may have noticed that there are a number of new appendices in this version. For a long time, we've only had the one appendix, and our growth just exploded out in this area. We'll go over each appendix and we'll cover the content of each and some of the background. Our first new appendix is Appendix B, Trial Summary Parameters for Submissions for Clinical. There are 60 new parameters. Well, they're not new parameters. There are 60 parameters indicated in this appendix. And these all use CDESK control terminology, and these are parameters that describe a trial in such a way that all FDA reviewers would want to see for any kind of trial. That is if that parameter makes sense for the trial that you're submitting to the agency. So if that parameter makes sense, please include that parameter, please populate a value for that parameter, and otherwise follow the appropriate implementation guide for your overall submission. For Appendix C, it's much like Appendix B, only these are the parameters that we'd like to see for non-clinical. There are 41 parameters in this table. Each of these parameters are already using existing CDESK control terminology. And these parameters are also desired if it makes sense for your trial. So if these parameters apply to your trial, please populate that parameter and give a value for that parameter, and otherwise follow the appropriate implementation guide for your submission. Appendix D, these are additional documents that have been evaluated by FDA. There are a number of documents put out by any standards data organization that are not appropriate to be included on the Data Standards Catalog because they may not be a data standard or a complete standard. So for those, we still would like to give comment to industry on how it may or may not be appropriate, or if it fits with our review needs. So we've developed this appendix, Appendix D, to give our comment that we encourage you in the industry to use this document as you put your data together and you would submit it to the FDA. So, for example, we have
Hello everyone. Apologies for the audio issues. We're getting that squared away.
looks like we have a clear. And should I start with the current slide?
Yes.
All right. So we'll just do this again. Some of you guys may have heard this. It's a repeat because we had some audio issues. Appendix D, which are additional documents that have been evaluated by FDA. The background here is that there are many documents put out by specific standards data organizations that are not actual standards or not complete standards. And so we can't put them on the Data Standards Catalog, but we still want to give comment to industry about if we'd like to see data submitted in a way that conforms to the content of these documents. So it's been a known gap for a while for us at the agency, and we found a way to communicate to you the appropriateness to use these specific documents listed here in this appendix. So the first document now listed is the Confirmed Data Endpoints for Exchange, otherwise known as Codex, for Send IG version 3.0. So you guys can use that for instruction or for implementation for your data. And we'll continue to evaluate these documents and we'll communicate to you about them here at this particular appendix. Appendix E is example study data folder structure. We thought we'd give you guys some more detail on the ECTD folder structure and where specific things would go in that structure. And as those details may change or arise, we'll continue to communicate to you about those here in this appendix. You can see specifically, but this is a detailed example of file folder structures for non-clinical data sets in both standardized and legacy formats. You can see they're called, each of the folders is called out there on the right-hand side of the graphic. And specifically, even when send is required and submitted, the tumor XPT data set should be placed in that current folder structure, analysis/dataset/legacy. You can see the arrow there pointing down there to the legacy folder within the ECTD structure. For more details about that, or for reference, you can see section 4.1.3.3 of the current technical conformance guide. Appendix F, we have an example of a simplified trial summary data set for clinical data. This is that one-line trial data set that is used to establish a study start date. It's 3 variables, and here's the specific values to populate those variables, and here's an example for industry you can simply cut and paste and use in your data preparations. Appendix G is example of simplified trial summary data set for non-clinical data. And again, this data set, this one-line trial summary, is used for the sole purpose of establishing a study start date. Here are the 3 variables, and here's how those variables should be populated. So we've cleared all of the appendices. We'll go on to some other new sections that we have. In the TCG, in section 4.1.1.3, we list some new parameter codes. These parameter codes allow the agency to understand which therapeutic area user guides, otherwise known as TAUGs, technical specifications, implementation guides, and models were utilized for a particular study. The agency worked with EVS for these terminology extensions for the variables TS-PARM and TS-PARM-CD, and a complete table of each of these variables with examples is in section 4.1.1.3. Section 5.2 is our Therapeutic Area User Guide, otherwise known as TAUGs. We added version numbers to the TAUGs. When we first wrote our first TCG back in 2014, there was only one version of any of these TAUGs, and the developers have been busy, so we found it expedient to refer to these TAUGs by version, and you will see that in section 5.2. And we've also added the trial summary parameters to capture the use of these TAUGs as we went over in the previous slide. More about section 5.2 Therapeutic Area User Guides, also known as TAUGs. We have some new ones for the October 2018 TCG. These are the Major Depressive Disorder Therapeutic Area User Guide version 1.0, Duchenne's Muscular Dystrophy Therapeutic Area User Guide version 1.0, Traumatic Brain Injury Therapeutic Area User Guide version 1.0, and Vaccines Therapeutic Area User Guide version 1.1. Also in section 4.1.1.3, we cover protocol deviations in otherwise the DV domain, which are protocol deviations. The DV domain should be included in your submission. It will be used by reviewers to examine protocol deviation trends of various study sites in order to facilitate the bioresearch monitoring program, otherwise known as BIMO, Clinical Investigator Site Selection Process. And once FDA tools are developed to extract and format needed data from SDTM to populate line listings used by the Office of Regulatory Affairs, ORA investigators during inspections. And the following variables besides the Cdisk required variables should be included in the DV or deviations domain when submitting DV data. And there's the list of the variables there. You can see them and reference them in section 4.1.1.3. And now we'll have some send updates from Dr. Thompson.
Hello, I'm going to talk about the updates to the non-clinical CDESK Send Data Standards section of the Technical Conformance Guide. So the send updates that I'm going to be talking about are where tumor.xpt should be placed, some clarification, about including the simplified ts.xpt with tumor.xpt. It should be placed in the same folder. Some detail about constructing the LB domain for send and how to place categorical data. Some detailed instructions for representing the lower limit of quantitation. The body weight gain domain is not required for CDER submissions, and how to limit your comments domain. We're also, as Helena mentioned, we've placed the codex, which explains the data that can be confidently exchanged in the SEND standard in our appendix. And so we're referencing that now and encouraging you to use it. So when SEND is required and submitted, tumor.xpt should still be included for rodent carcinogenicity studies. And when tumor.xpt is submitted, it should be placed in the analysis part of the ECTD in Analysis Dataset Legacy. And figure 4.1.3.3 in the TCG gives a graphical representation of where that data goes. For the LB domain, we've provided some clarification about how to use the SDTM and SEND model to represent data for categorical data. Some lab tests, like urinalysis dipsticks, essentially give categories, even though those categories might have a number. So you could have a result of negative, 100, 250, 500, but you haven't really measured anything. It's just that the numbers are the categories. So it's important not to make the mistake of treating those as numeric data, but instead to treat them as character data. So all of the results for a test that gives categorical data should be placed in the stress C variable, and stress N should be left null. And what this allows FDA to do is to create incidence tables automatically by counting things up over the stress C variable, and rather than creating inappropriate averages and standard deviations and all from categories. Additionally, also in the SEND LB domain, sometimes a particular number is used to represent the lower limit of quantitation in the averages in the non-clinical study report. And it can be very confusing to try to understand the provenance of the averages in the study report if the number in the limit of quantitation is not placed somewhere in the SEND data set. So if you used a number to create group means in a test that was outside the limit of quantitation, that should go into SUP LB and Lab Calc N. And this is really only for the specific case where you had measurements outside the limit of quantitation, for example, below, and then you used a number to calculate the averages. We've also added some more information to the PC domain directions, and these are primarily to help people understand what the SEND IG says about handling results that are less than the limit of quantitation. So we found it fairly complicated to use all of the different implementations of SEND that we're receiving. So we've put these detailed bullet points that are at the top of the slide into the TCG. I'm not going to explain them in detail on the webinar. I would instead prefer that you refer to section 4.1.3.3 and take your time to look over them yourself. With the BG domain, for CDER submissions, and CDER right now is the only center that is requiring send with certain non-clinical studies, you do not need to include a BG domain with your send submission. You only need to construct the BW domain. This is specifically for CDER, and other centers are not requiring send and haven't looked at the body weight gain domain requirements. When you include a comments domain in send, it's important to limit the comments that you include to those that are actually relevant to the study interpretation. Review software that we use to display information to reviewers doesn't have a way of discriminating between small pieces of information that were captured in a non-clinical LIMS, like somebody walking into a room and noticing something about an animal or an observation that wasn't captured in the actual send data set, and between important pieces of information that would be relevant to study interpretation. So we're asking you to try to limit the comments to those that are relevant to the study so that the reviewer doesn't have to click on every single comment and go down the list and try to figure out what you thought was important and included. So at this point, and this is our additional resources with links into the study data resources and technical conformance guide. So I'm going to turn things back over to Ray, and he's going to wrap up the seminar portion of today's webinar.
This is Ray Ford, and we'll now transition into our Q&A session. We invite you to continue submitting your questions. We'll begin in a few
Hi everyone, we're back. Thank you for your patience while we went through your questions. We had quite a few here, so we'll just get started. Here's a question, I'll read it. Question about the 60 parameters for TS, that's trial summary, for clinical studies. Should all 60 parameters be included in the data set with a null flavor for ones that aren't applicable? At this time, I would encourage you to use only trial summary parameters that are applicable to your study, though the null flavor, the TS and val F variable certainly is implementable in the CDER standard. So we're not going to tell you to not implement something to deviate from an implementation guide. Next question. Dear FDA, I would like to have more detail on the update to the DM demographics domain in section 4.1.1.3 SDTM domain specifications. It states, additional enrollments / screenings should be included in a custom domain with a similar structure to DM. 1. What variables should we include? Mainly USubjID / SubjID and site ID, comma, investigator ID, comma, investigator name, comma, country, if necessary due to a different site being used by the subject? Or should we include all the required and expected DM variables, example, the reference dates, age, sex, ARMCD, etc.? Do you have a domain abbreviation you would like us to use, example, DQ, which the SDS team was discussing using? That's a lot of question. So I'll start with question number 1, which is what variables should we include? You should include the variables that follow your implementation guide that you're implementing, that makes sense for your submission. And if you have questions about a specific submission or have a specific use case that you need more details on, I invite you to send a question to eData. The 2 addresses are up right now on the screen. For questions to CDER, the email address is cder/edata@fda.hhs.gov. And for additional questions to CBER, that email is cber.cdisk@fda.hhs.gov. The second part of that question says, do you have a domain abbreviation that you would like us to use? And the agency has no opinion on that at this time. Do we have any other questions? I have one here. So we are getting a lot of questions that are taking the content for the LB domain, the laboratory data domain that we gave details for in the send update, the send portion of this, and ask about that requirements to SDTM. We did not go over any of the SDTM updates for the LB domain in this webinar in the interest of time, so I invite all of you to go to the specific sections and general consideration sections for both SDTM and send to look at the specific updates there and to reinforce what you heard from Dr. Thompson today, and also give the details and our requirements, or our expectations, not requirements, for the LB domain and SDTM. And if you have any additional questions or need any confusion or have any confusion about after reading in the TCG, again, I refer you to eData. We're really good about answering those questions, even though sometimes it can take a little while. Okay, we have some send questions.
So the first question is, which Module 4 sections require send non-clinical data sets? And the current send requirements are for single-dose toxicology, repeat-dose toxicology, and carcinogenicity. Those are usually placed in 4.2.3.1, 4.2.3.2, and 4.2.3.4 in the ECTD. someone has noticed that the study start date is not listed in the trial summary parameters list for clinical trials. Yes, please do include the study start date, STSTDTC, in the trial summary dataset for clinical trials. Who academic medical centers, in parentheses, is required to utilize the Study Data Technical Conformance. The Technical Conformance Guide applies to all sponsors and applicants who are submitting commercial applications. We've gotten a couple of questions about the arrow to analysis data legacy. Looks like it was pointing to tabulation. Is the arrow correct? Refer to the Technical Conformance Guide. We had some conversion issues from PowerPoint to Adobe, and I think that arrow moved. So please refer to the actual Technical Conformance Guide for the placement of the data sets. Somebody noticed that there was a trial summary parameter code, ARM code, that was included in the send list. We've already made the corrections to the Technical Conformance Guide, and we removed ARM code from the non-clinical trial summary. So please make sure that you have downloaded the latest version of the Technical Conformance Guide when you refer to it. When submitting a trial summary.xpt for legacy non-clinical data, should a defined file be provided as well? Yes. For legacy non-clinical data, which is either a tumor.xpt or data sets for animal rule studies, reviewers have continued to request a define.pdf file that helps them to understand what tables, variables, and values you have provided in the data sets. So please continue to submit both the abbreviated trial summary.xpt and a define.pdf. Final question I'm going to address in this round are, if a QA audited draft report for repeat dose tox is being submitted with an initial IND, are send data sets required at that time, or can they be submitted later with the final report? We understand that this is a bit of a pain point, but the non-clinical reviewers do use the send data sets. So they have requested that when a draft report is submitted with an IND and the 30-day clock starts, that they are also provided with the send data sets so that they can actually make use of them during the review.
Okay. Hi, folks. We've gotten a number of questions about the analysis results metadata, or ARM, which is a CDISC document. At the time, the agency does not have a position on this document, but if we were to have one, we would put it in the appropriate Appendix D, where we currently put the codex document. So if you have, so check back to that specific appendix, and you'll have our thoughts and opinions on those documents. Hello, everyone. We're still looking at your questions. We'll have a couple of more. We're just getting it together. We'll get back to you in just a second.
Okay, I have a couple more send-related questions, or this is sort of. So in SDTM LB for capturing LOQ, should they be derived at ATOM or SEP-LB Lab Calc N? And in SDTM, you're going to continue your normal practice of using ATOM datasets for derived information. SEND does not have ATOM, so we rely more heavily on specific directions of SUPs and implementations, and that's why we're also putting specific directions about SEND in the SEND section, specific directions about SDTM in the SDTM section, and information that applies to both standards in a both section. So we place these instructions very deliberately to apply to the specific standard. And the other question is, how does one determine if a pilot study goes into 4.2.3.2 versus 4.2.3.6, i.e. plus or minus SEND? The SEND requirement is based on study types, repeat dose, single dose, carcinogenicity, and certain types of safety pharmacology once SEND 3.1 is required. This is not specific to ECTD placement, it's specific to study type. And if you have any further questions, then please remember to ask your review division, because the reviewers are the ones who use the data and are the end consumers. Okay, for a vaccine being developed under the animal rule, is it worthwhile to include non-clinical studies that are outside the scope of the current FDA Data Standards Catalog in the SDSP? So yes, the reviewers use the SDSP. So if you're submitting data sets, you can make use of that document template in your pre-submission conversations with FDA, and you will almost always have some pre-sub conversations with FDA as you develop your data plan, even if the studies do not require standardized data. So then the SDSP is a very good place to use to document the outcome of those conversations and communicate to the reviewer what you're providing. And the last question I'll address is, are we requested to use the 60 new TS parameters for new studies only, or for the next submission even when it is next month? We understand that the technical conformance guide comes out and you can't simply flip a switch and implement things immediately. So what we'd ask is that you begin your implementation now, and as soon as you're ready to submit all 60 TS parameters, you begin doing so. They're of very high value to the agency and reviewers, and most of those parameters were in the old version of the FDA business rules. So we expect that most people will be ready for most of the parameters, and then please add them reasonably. We would prefer not to have you submitting a year from now and not including all 60, but if it's next month and it can't happen, we understand. And I'm going to turn this over to Ray now to wrap up.
Thank you, Helena and Elaine, for your very informative and timely talk, and also for our panel for responding for the questions that came in. A few closing reminders. This activity has been pre-approved by RAPS, is eligible for up to 1 credit towards a participant's RAC recertification upon full completion, and SQA approved for 0.25 non-GCP or non-GLP units per 1 hour towards RQAP re-registration. You'll be able to obtain the attendance certificate only upon full completion of the survey, which will remain open for 2 weeks. Let us know your comments and feedback via the survey. This will provide us with ideas for future improvements. On behalf of CDER Small Business and Industry Assistance, we hope that you enjoyed today's presentation and found it helpful. We look forward to your presence at future webinars, which will be advertised on our website at fda.gov/CDER SBIA webinars. Thank you.